Hello, and welcome to another exciting episode of Edpuzzle Notes. This is the end of cellular respiration. Now, you've seen this before, electron transport chain. This was photosynthesis. We're going to go over the electron transport chain in respiration here in a second, but I just wanted to show you that it's basically the exact opposite of this. In photosynthesis, we had water, this Mickey Mouse, donating its electron into photosystem 2 there, and then it got split into its two hydrogen ions and its oxygen. Well, two of those oxygens bonded together and they would leave the thylakoid and float out into the air. This is going to be the exact opposite. So O2 is not going to leave. This time oxygen is actually going to be the one hanging around here and is going to grab those electrons. Now, instead of blowing up this Mickey Mouse right here, instead of splitting the water, the O2, when it gains these electrons, is actually going to grab two hydrogens, and we're actually going to make water in respiration. So in photosynthesis, we split water to get the electrons. In respiration, we actually make water. When oxygen grabs the electrons at the end of the chain, bonds with two hydrogen ions to form H2O, or water. If you remember, when it got over here to cytochrome, in photosynthesis, the energy of these electrons was used to pump hydrogen in. Well, same thing, but except in respiration, it's going to be the opposite. The energy from those electrons is going to be used to pump hydrogen out. When we got to the final step, Mr. NADP was waiting to grab these electrons to form NADPH. Well, it's going to be the opposite of that in respiration. NADH and its buddy FADH2, they're the ones giving up electrons into the electron transport chain. So it's basically the exact opposite. All right, so we've already seen this slide. We've gone through the first three reactions in respiration. We got 2 ATPs in glycolysis. We made 2 ATPs in the Krebs or citric acid cycle. So far we've made 4. In the first 3 processes or reactions, we've made 10 NADHs. Each one of those 10 NADHs is they're all carrying 2 electrons and of course a hydrogen. We also made 2 FADH2s. Remember, these guys also, just like its buddy NADH, they carry electrons. They carry 2 electrons. And we've made all of the CO2 we're going to make. 6 CO2s, because that's in our respiration formula. So we're done making CO2s. Now we're going to use these electrons. We're going to use all these electrons that we've acquired in the first 3 steps. We're going to use those to make more ATP, a lot more. All right, so everybody should remember what this organelle is right here. That is a mitochondrion. Mitochondrion, the powerhouse of the cell, because that is where most of cellular respiration takes place, other than glycolysis. All the remaining 3 reactions all take place in the mitochondrion. Remember, the first 2 reactions inside the mitochondrion take place in what's called the matrix. That's just like the middle of the mitochondrion. If you look, the mitochondrion has 2 membranes. So it's got one here on the very outside, and it's got another one here on the inside. This little membrane that's like right there. It's hard to see it, but that's where the electron transport chain is going to take place, in this inner membrane, the inside membrane. All right, so here is the electron transport chain. Basically, this is what you need to know. So, important notes, you don't need to write that. But in this reaction, we're going to use those high energy electrons that are being carried, of course, by the 2 molecules, NADH and FADH2. Remember, all we need to know is those guys carry electrons. Now we're going to use those electrons to make a whopping roughly 28 ATPs. We're going to do that during what's called the electron transport chain. And you saw it earlier in the photosynthesis. It's basically the same exact thing as photosynthesis, just in reverse, backwards. One thing you'll need to know, oxygen. This is why you have to breathe oxygen. You're probably, wait, why do I have to breathe air? What do I need it for? Well, oxygen serves as the final electron acceptor. Basically, oxygen sits at the end of the electron transport chain, and when those electrons are done using their energy, oxygen grabs those electrons. It also grabs two H+, two hydrogens, and one oxygen plus two hydrogens makes water. And that's where we're actually gonna make 6 water molecules during the electron transport chain, along with 28 ATPs. All right, so here's the electron transport chain in respiration. You really don't need to take many notes on this. This is not going to be an essay question like there was an extra credit essay question in photosynthesis. There will not be one here. Some people just like to know how things work, and this is kind of how it works. All right, this right here, we're just going to call him protein complex number 1. He has a very long name, but we don't need to know it. This is where NADH drops off its electrons. Electrons. And for every electron that it gets dropped off, 1 hydrogen ion is going to be pumped out. So it drops off 2 electrons. That's going to cause 2 hydrogen ions, H+, hard to write, to be pumped out. This complex 2, this is where FADH2 drops off its 2 electrons. It gets passed to this mobile carrier and then passed on to complex 3. When the electrons reach here, 2 more hydrogen ions are going to be pumped out. Outside, this is the inner membrane of the mitochondria, and this is between the outer membrane and the inner membrane, so it's being passed into this outer membrane space. It then gets passed to cytochrome C, which takes it to the 4th complex, which also pumps 2 more hydrogen ions outside. So we're getting a high concentration of hydrogen ions out here. This is where also oxygen, O2, is just hanging around. When those electrons get done pumping out their hydrogen ions, the electrons join with oxygen, and when you add 2 hydrogens to it, you get H2O. So this is how water is produced. Oxygen serves as what's called the final electron acceptor, which just means oxygen takes the electrons when they're done going through the electron transport chain. So the energy from those electrons was used to pump all of these hydrogen ions outside the inner membrane. So now we have a high concentration of hydrogen ions outside. We got a low concentration on the inside. Well, just like in photosynthesis, these hydrogen ions all want to go from high to low. And they all diffuse through this protein. He has the same name as it had in photosynthesis, ATP synthase. And that movement of hydrogen ions from outside the inner membrane to inside is used to convert ADP into ATP. All right, now this will be super important. It basically just shows you everything we've done so far. We started off with the first reaction called glycolysis. Remember, that occurs in what's called the cytoplasm of the cell, basically just the fluid that's not within an organelle like here, the mitochondrion. In glycolysis, we made 2 ATPs. We also made 2 NADHs, which go all the way over to the electron transport chain. In our second reaction, called pyruvate conversion, we made 2 acetyl-CoAs. We made 2 NADHs. Now, it doesn't say it here, but we also made 2 CO2s as a waste product. In the third step, which also occurs in the mitochondria matrix, just like pyruvate conversion, we made 6 NADHs. which carry high energy electrons. Remember, these guys carry electrons. We made 2 FADH2s, and those were going to be used in the electron transport chain. If you remember, we also made 4 CO2 during the citric acid or Krebs cycle, and we made 2 more ATPs. And the electron transport chain, it says 34. It used to be about 3-4 years ago. Not anymore. I'm going to scratch that out. It's about 28 ATPs in the electron transport chain. So we do not make a maximum of 38. We make usually roughly around 32 ATPs. You will not need to know that number precisely. On a question on the test, it would be something like this. During aerobic respiration, how many ATPs are produced? A, 2, that's false. B, 8, false. C, 32. D, 150. Obviously, 32. Somewhere in the 30s, 32, 34, 36. Just know it's somewhere in the low to mid 30s is what we make in aerobic respiration. All right, now really quickly, we're going to go over anaerobic respiration, which we remember it's energy that is produced without oxygen. So no oxygen. That's anaerobic. If it's with oxygen, that's aerobic. We also call it fermentation. It's another word for anaerobic respiration. So these things basically mean the same things. In our cells, you're an animal, in case you didn't know that, it's called lactic acid fermentation. The reason for that is if you lift weights really, really hard and there's not enough oxygen present for your cells to use aerobic respiration, they start going anaerobic. And when they do that, you produce something in your muscles called lactic acid. And in case you're curious, that's what makes you feel sore. But the main thing you need to know about this, in anaerobic respiration, we do not make all the ATP. We don't make 32 ATPs like we do in aerobic. We make a measly 2 ATPs. So know that. Anaerobic respiration, you only get 2 ATPs. All right, another type of fermentation. So in your cells, you guys perform lactic acid fermentation. In some single-celled organisms, like yeast, they call it alcoholic fermentation. And it's basically pretty close to the same process. They still make a whopping 2 ATPs, which isn't very much. But they also make, instead of making lactic acid, which makes our muscles sore, yeast make alcohol or ethanol. Ethyl alcohol, that's just the alcohol that people that are above 21, some people drink that stuff. And that's how it's produced, through alcoholic fermentation. Glucose is the storing of glucose. In plants, they store it in 2 ways. They either store it in starch. That is just a carbohydrate. So if you eat bread, if you eat potatoes, that is filled with starch. They also can store it in oils. So I'm sure if you ever cook with vegetable oil, canola oil, that is plants storing glucose in their oils. So plants can store it in starch or oils. Animals, we store it in different ways. We store it in a larger sugar molecule called glycogen. And of course, we can also store it, some of us have it around our midsection, I do. You can store it in fat. So animals store it in glycogen or fat. All right, the last slide. This is going to explain why anaerobic respiration is important. Regular bios, you don't need to take a lot of notes on this because I don't even know if it's going to be a question, to be honest with you. Honors, you might want to know it, just the basics of why it's important. And the reason is, if you remember, well, first of all, without anaerobic respiration, all energy would stop. If there's not enough oxygen present, all energy production would stop. The production of ATPs would totally shut down. But with anaerobic respiration, we can continue to make the 2 ATPs we make through glycolysis, anaerobic. Well, the reason for that is in glycolysis we need these dead batteries. We need these NADs. Well, the NADs are made when NADH drops off its electrons in the electron transport chain to form NAD. Well, you can't make these NADs in the electron transport chain unless there's oxygen. So without oxygen, we're going to run out of these NADs. And we need them because every time glucose gets split, we're freeing up electrons in the glucose. So we need these NADs to come and grab 2 electrons, and so we don't end up with a pile of electrons here. We can't have a pile of electrons just sitting there. So we need these NADs to grab the electrons, become an NADH. Well, if there's no oxygen, there's not going to be any NADs coming from the electron transport chain. So what happens is, just like regular, we make 2 pyruvates in regular glycolysis, but instead of this pyruvate going off to pyruvate conversion and then the Krebs or citric acid cycle, energy is actually used. 2 NADHs are actually used to convert pyruvate into lactic acid. And the reason that's important, when we burn these 2 NADHs, we get 2 of these dead batteries, these 2 NADs, which can now pick up some more electrons. And it just goes around and around and around. We use NADH to turn pyruvate into lactic acid, and then we can get 2 more of these dead batteries to pick up the electrons that we keep getting when we split glucose. Thank you, and I hope you enjoyed it.